There are three types of cell receptors. These include ion channel receptors, G-protein coupled receptors, and tyrosine kinase receptors. The first we will cover are the ion channel receptors. These are transmembrane proteins, meaning they span both sides of the cell membrane. These proteins transport ions into the cell. They include a signal binding site, a gate, and a transmembrane space. A ligand will activate the protein and open the gate by attaching to the signal binding site, as we can see in this next step. This allows the ion channel to be opened and ions to enter the cell. Finally, the ligand detaches from the binding site and the gate closes, meaning no more ions will enter the cell through the channel. The next type of receptor that we will cover is the G-protein coupled receptor. This receptor is made up of three parts: a transmembrane protein with a signal binding site, a G-protein, and an internal peripheral enzyme. And of course, there's the ligand. When this ligand attaches to the signal binding site, the GDP attached to the G-protein is replaced with a GTP. This activates the G-protein, which moves laterally along the interior cell membrane to an enzyme, activating that enzyme through phosphorylation. Once this occurs, the ligand detaches from the binding site, and the G-protein loses its GTP and regains its GDP, resetting the G-protein coupled receptor back to its inactive state. Finally, we have tyrosine kinase receptors. A single tyrosine kinase receptor involves two transmembrane proteins which have some tyrosines on their inner membrane sides. Once ligands attach to the signal binding sites, the two transmembrane proteins dimerize or come together. Once dimerized, the tyrosines that were previously mentioned are phosphorylated by ATP, which in turn phosphorylates enzymes which can then begin a phosphorylation cascade, activating the signaling pathway. Finally, an enzyme called dephosphorylase removes the inorganic phosphates from the tyrosines, and the receptors separate from one another once the ligands detach from the signal binding sites, which finally returns the receptor to its original state.